Thank you, Alexander. Alrighty. Would you like to kick us off, Dr. Pepe?
Welcome and good afternoon. I'm Pei Ping from the UCLA-based Bridge to AI TRM and Bridge to AI Training Center. On behalf of Dr. Carol Watson, we thank you all for joining us on this important lecture from our partner Bridge to AI Grant Challenge team to enrich the Bridge to AI consortium-wide learning resources.
I'm Jack Rincon, a Bridge to AI TRM Working Group Project Manager, working with Dr. Pepe Ping. It's my pleasure to co-host this session and introduce the next TRM lecture from the Functional Genomics Grand Challenge dubbed CM4AI.
Thank you, Jack. I would like to do a brief reminder to our audience. This is part of our lecture series in providing insights into databases across Bridge to AI consortium initiatives generated on a Grand Challenge specific topic. In this particular case, our goal is to prepare interested stakeholders, both within our consortium as well as in the broad scientific community, to engage with Bridge to AI-ready biomedical datasets to gain better understanding of the dataset structure and their potential applications.
The CM4AI project is set on a mission to map the spatiotemporal architecture of human cells for interpretable genotype-phenotype learning. This initiative addresses the challenge of machine learning models in genomics, often functioning as black boxes by employing proteomic mass spectrometry, cellular imaging, and CRISPR-Cas9-based genetic perturbation. These approaches aim to create a comprehensive library of cellular structure and function maps across diverse demographics and disease contexts to address precision medicine.
I would like to invite our audience to join me to welcome the renowned speakers of today's lecture. First, Professor Trey Ideker, a world-renowned professor of medicine, bioengineering, and computer science from UC San Diego. He has been leader and a pioneer in system biology approaches to omics science and to create AI-ready datasets. As the lead for the Bridge to AI CM4AI project, he has been working to successfully mapping molecular networks as well as integrating protein datasets, imaging datasets to advance AI-driven precision medicine. The work his team has done is being viewed as groundbreaking in the community, and it has shaped the fields like cancer research and neurological disorders. He is widely regarded and highly respected using bioinformatic tools to create a number of resources for Bridge to AI. Back to you, Jack.
Thank you, Pepe. Then we have Dr. Tim Clark giving us details on data preparation, releases, availability, and standards. Dr. Tim Clark is an associate professor at the University of Virginia and a leader in biomedical informatics and AI readiness. He leads the standards module for CM4AI and co-chairs the standards working group for Bridge to AI, where he developed the FAIRscape framework to ensure ethical and robust AI data analytics. His expertise spans functional genomics, predictive analytics, and cognitive disorders, making him an essential figure in preparing CM4AI data for impactful research. Next, we have Danielle Pacia, a research associate at a leading bioethics think tank, the Hastings Center. Danielle's work focuses on ethical, legal, and social implications of AI and biomedical research, including the ethical challenges posed by initiatives like CM4AI. With a master's in bioethics from Harvard Medical School Danielle's insights will guide us in addressing the complex ethical considerations surrounding AI applications in precision medicine. And finally, we finished strong with Dr. Wade Schulz, an assistant professor at Yale School of Medicine and an expert in computational healthcare and data science. Dr. Schulz leads efforts in managing large biomedical data sets and predictive modeling, bringing his experience as director of informatics at Yale to the Bridge to AI initiative. His work includes fostering collaborations and partnerships to enhance CM4AI's mission of addressing AI-driven biomedical research.
With this team of distinct speakers, we're hoping you will gain an understanding of the mission and scientific goals of the Functional Genomics Grand Challenge projects at Bridge to AI. Learn about their datasets creation protocols, their unique ethical considerations, and explore opportunities for collaborations within the Bridge to AI consortium and beyond. With that, I will hand it over to our esteemed speakers. Trey, it's all yours.
Okay, great. Thank you very much, Pepe and Jack, for that great introduction. And we're now sharing the materials. Can everyone see those?
Not quite.
Let me know when you can see it.
We're not able to see anything at the moment. If not, Jillian, you can just share your screen. You had success earlier.
Yeah, we can see it actually. Danielle says she can see it as well.
I can see it. Yeah.
Okay, so maybe Jack, it's just your end somehow. Whoever is recording, we should make sure they can see it.
I'm good. We're good.
Okay, fantastic. Okay, great. Well, thanks again, Jack and Pepe, for that great introduction. It's my pleasure to introduce the Functional Genomics Grand Challenge, which we call Cell Maps for Artificial Intelligence, or CM4AI. As you can see down here at the lower left hand on my screen, this project involves 9 collaborative institutions, and several of which are represented by the speakers today. Next slide, please. So the point of departure of our grand challenge is really the point of departure for all of human genomics as it intersects with artificial intelligence and the opportunities for artificial intelligence in understanding the human genome. As we all know, AI has revolutionized many tasks, both in and out of biology and medicine. And in particular, the question that concerns us here for functional genomics is, how does the genetic code dictate the architecture of living systems, number one? And number two, how do changes to that code promote or prevent risk for disease? If you think about those two questions as an input-output diagram, as one often does in artificial intelligence machine learning, at the very highest level, on the left, we have an individual's genome sequence. We may have other information about that person or patient we wish to express, such as their clinical history. And we'd like to translate patterns in the genome sequence and other information we have for the patient into, first of all, changes or impacts on the molecular structure and function of that person's cells and tissues. And second of all, how changes to that molecular structure and function predict various phenotypic outcomes, predicted behaviors, risks for disease, treatment response in the case of precision medicine. And I'll use that last example. Thank you for the slide advancement. I'll use that last example to give a pictorial view of what this actually looks like. That's the schematic diagram, but in reality, we're going for systems such as shown here that would be embedded in your phone or other handheld device or desktop device, to which you would ask questions. And so in the case of precision oncology, you might want to say, "Dear AI, my patient has just been genome sequenced for their tumor mutations, and we've discovered the following 50 mutations in different genes, such as PTEN and DAM1 and 48 others. Key questions would be, what drug should I give her? That's if you're in a clinic. If you're in a pharmaceutical company, a key question might be, should I be looking at any new drug targets? And so on and so forth. Next slide, please. And so this leads us to the focus of the CM4AI Bridge to AI data generation focus, which is, how do we get information about that molecular structure and function that's important for both healthy function and diseases like cancer to feed to the AI system? Where does it get that knowledge? In the case of large language models get their knowledge by reading documents on the web. But how does it read or otherwise learn about the structure and function of cells and tissues, particularly because there's so much that's not yet known about that structure and function? That essentially is the focus of this project. Next slide, please. And so if you think about the human genome itself, as has been oft presented as a parts list of what are the genes encoded by each of our cells, shown here, the analogy, one of the many analogies that can be drawn as to the parts of a car, here a Volkswagen Golf. Those parts, of course, fit together and play together in molecular assemblies and pathways. And it's that wiring diagram that one really needs to know, and the AI will ultimately need to know if you're to make inferences about the human genome and how different variants and mutations in that genome lead to drug responses and disease states. And so really, what is that? Sorry, not yet. So what does that parts manual look like for a car? Well, it's the workshop manual, like shown at the lower left, that we then read, which tells us how to assemble those parts into the full car. That's the level of wiring diagram or assembly manual that we're looking at for the human genome and the cells it encodes. Next slide, please. So now coming down from the 30,000-foot view, somewhat lower, this is the end-to-end pipeline that really defines the CM4AI project. We have taken a trifold approach to generate data that informs how cells are assembled from their component genes and proteins. Those data sets I'll talk about in just a moment. And those three data sets are then integrated by AI approaches and visualization approaches into maps of human cell architecture. If you think about what are the scale of those maps, they span the scale of nucleotides in the nucleus of about 10 to the minus 9 meters, all the way up to the whole size of a human cell at about 10 to the minus 5 meters. And it's then these maps that the CM4AI project provides to downstream AI ML applications of all kinds. So there's many tasks that these masks are these maps will be embedded within. Next slide, please. To accomplish this cell mapping pipeline, we've assembled as I mentioned, 9 different institutions, and here are some of the leaders at those different institutions who are enabling these cell maps and their utility in machine learning applications. So focus 1, or overarching goal 1, is enabled by Emma Lundberg, Nevin Krogan, and Prashant Mali, who are generating the 3 major data types for cell mapping. Project or goal 2 is led by Tim Clark. You'll hear from him just in a moment, which is enabling AI-ready integrative cell maps to be constructed and well documented using those 3 data types. Project or goal 3 relates to disseminating the maps and putting them in the hands of both the tools and the people who can best benefit. And then goal 4 relates to education, ethics, and engaging our stakeholders. And you'll hear from Wade Schulz, who's the leader of that goal in just a moment, as well as Danny to talk about the ethics. So could you please go to the next slide? So let me just, before I turn it over to Tim, take you through what are the 3 data sets that we're generating in this project, and what are the cell models that we're trying to map Well, those are twofold for the first couple of years of the project. We began with a triple negative breast cancer cell line, and now we've transitioned fully to our main targeted cell line for this project, the human-induced pluripotent stem cell, which is the progenitor of all cell types. So they're therefore a great place to start mapping. And in each case, we'll look at the naive cell population. In the case of breast cancer, we'll also look at that how that cell is remodeled and how that map changes in response to two different standard of care drugs, paclitaxel and vorinostat. In the case of the iPSCs, we'll look at their naive, undifferentiated or embryonic cell state, in addition to differentiated neurons and differentiated cardiac cells. We will then interrogate all 6 of those cell states. If you think about those cell models as basically defining 6 points of a triangle, as shown over there at left, we'll interrogate those with 3 different experimental platforms. First, confocal protein microscopy to look at the distribution of large numbers of proteins inside of cells in terms of subcellular compartments and smaller protein assemblies in which they localize. Number 2, we'll use a technique known as affinity purification tandem mass spectrometry, or APMS, to read out what are the immediate interacting partners that biophysically interact with each of those proteins. And then 3, is a functional measurement called Perturb-seq, which uses single-cell RNA sequencing to characterize the transcriptional response to knocking down, editing down each of those proteins with CRISPR technologies. Next slide, please. And all 3 of these technologies get put together into cell maps. And I'll just give you a brief overview of what one of those maps looks like. This is a hierarchical view of the map, and so we start at the top with the collection of all proteins in the cell, and that's what the root of that hierarchy looks like. And then based on the 3 data types, or subsets of those 3 data types, we can now, at a high level, fractionate the proteome into mitochondria, nuclei, cytosol, microtubules, and so on. But then you don't stop there. You continue this iterative procedure of further fractionating the proteome into smaller and smaller compartments, all the way down here. So you see here underneath the nucleus, we can resolve nuclear speckles. In fact, there's 14 kinds of nuclear puncta, or nuclear speckles, that can be resolved by integration of imaging with the mass spectrometry data. And each of these nodes in this hierarchy below the nuclear speckles represents that. Each of these nodes is then a set of proteins, which is ripe for further analysis via structural proteomics approaches. And so shown up here at the upper right is the so-called Lambtor complex, where our mapping has already been able to add new components to a known, very important complex in both normal cell metabolism and, it turns out, cancer. Next slide, please. And with that, I will turn it over to Dr. Tim Clark.
Hi, thanks. So, as scientific researchers, it's preparing data sets for AI. It's incumbent on us to be able to demonstrate that those data sets were prepared with some kind of understandable and explainable process, right? There's no hand waving allowed here. And this is called pre-model explainability, right? So when we're releasing this data, we make releases of the data sets via the Dataverse generalist repository periodically. And we release them under open source non-commercial license. Well, in early 2025, we'll make our next release, which will be much more substantial. And you see on the top right, there's our initial publication on this is in bioRxiv. The AI ready data have fair that is findable, accessible, interoperable, and reusable, richly descriptive metadata that really show you the process by which these data sets were obtained, and allow you to track the provenance back as far as possible. One of our goals in year 3 is to take the provenance in a very formal, machine readable way all the way back to the samples. And we have developed this package called Fairscape to provide a framework that automatically can, once you integrate it into your system, it can automatically capture and package this AI-ready metadata. And we also, as part of the standards working group, developed a set of AI readiness criteria and representatives from all the grant challenges, and they're evolving and they're domain specific. And the lower right, that's our recently released paper in bioRxiv on that topic, and that's scheduled for release to a peer-reviewed journal shortly. Next slide. So these are the key criteria, and they're kind of it's multidimensional, and they're kind of a ladder in a way. They escalate from fairness, which is a well-defined NIH promoted standard, through detailed provenance, which we use machine readable provenance graphs based on a World Wide Web Consortium standard, deep characterization of semantics, statistics, potential sources of bias, data quality, all the way up through describing the ethical acquisition and management and dissemination and so forth, how sustainable the data sets are, and how computable they are. Next slide. So this is a cartoon of the complex process by which we assemble these data sets and put the metadata onto them. So the immunofluorescence microscopy, the AP, the affinity purification mass spec, the perturb-seq scRNA-seq comes in from the data acquisition groups into a tools pipeline. These are all packaged with metadata. The pipeline is actually rather complex, and we then package these in a standard way called RO crates, research object crates, which package the metadata in a graph language with graph language called JSON LD, and send it to a server, which integrates the provenance graphs and all the metadata, and then pushes them to a generalist repository. Next. So this is a little zeroing in on how this works. We have a client package that where the data is collected. The metadata is described in a set of parameters sent to the client package. The data and metadata are packaged. They're sent up to the Fairscape server. This is a kind of a push button process, and we've implemented a push button upload to any selected Dataverse repository. So the server site generates persistent IDs, fair IDs. It unwraps and stores the RO crates, integrates the provenance graph, and provides a web GUI. Next slide. So again, as I said, the metadata plus the data are available from the Fairscape server in human readable or machine readable fashion via the web GUI or through an API. And the provenance graph is based on a vocabulary called evidence graph ontology based on W3C prov. This is just a little cartoon. It's actually a screenshot from the web GUI of one step in the process of producing our AI-ready data. Next slide. And what we have set as goals. In our AI readiness criteria paper, we describe, and I think for the first time, really fully describe what criteria the Bridge AI program believes are required for basically any AI-ready biomedical data. And the green line is the goal by the end of year 4. We're halfway through the project. We have a very long pipeline to traverse into 3 labs. And our goal is to push the blue characterization of this year out to the green. And we hope to make very significant progress next year by working down the pipelines into the labs and adding quality assessment and ensuring all the primary data sets are deposited in NIH-specific repositories. Next. And now I'll hand it over to Danny, who will talk about ethics.
Thank you, Dr. Clark. I will be talking today about the various kinds of ethical considerations associated with CM4AI, and I'll start with just a really basic, somewhat high-level definition of ethics, just because of the diversity of the audience. In research, an ethical consideration can be really variable. It can look like informed consent, but it can also look like explainability, transparency, and accessibility. And it's really important to hold all of these considerations together and to balance them. Broadly, ethical considerations are essentially evaluating moral implications associated with a decision, an action, a policy. So a really broad way of construing what ethical considerations are. For CM4AI in particular, we have a more specific definition that was part of an article titled Cell Maps for Artificial Intelligence, AI-Ready Maps of Human Cell Architecture from Disease-Relevant Cells, which is available online. Here we note that in terms of ethical considerations, we need to look at if the data set was obtained, validated, documented, licensed, and distributed ethically. We also need to consider ethical treatment of subjects, as well as proper non-biasing data analysis, scientific integrity of methods, conclusions, and publications. Here in the middle, this is just to point out sort of an adage within the field of ethics, and it's that what is ethical is not always legal, and vice versa. What is sometimes compliant or considered to be legal is not always necessarily ethical. So oftentimes, considering the ethics requires going beyond what is compliant or legal. We can think back to a lot of instances in history where something was indeed legal or compliant through regulatory standards, but it might not be considered to be ethical currently. And one note about ethics and basic science, oftentimes ethical considerations are talked about in the realm of human participants. That is really well trod in the bioethics literature. Less well trod is basic science research and ethical considerations associated with that. With this particular kind of research, it's especially important to deeply contemplate potential downstream effects with these seemingly non-value laden decisions, largely because it's somewhat less discussed within the ethics and LC literature. Next slide. Some really high-level points to consider about CM4AI and the ethics of it. Scientific validity can and should be considered a part of ethical research. It's ethical for research to be scientifically valid, and that point really needs to be emphasized in this particular project. And it's also really important that transparency is prioritized, harkening back to a lot of points that Dr. Clark made about explainability, which we'll get into later in the presentation. Next slide. Getting into the nitty-gritty and talking about cell line selection and the ethical considerations associated with it. There are, again, several different variables that need to be considered when weighing ethical considerations. Specifically with cell line selection in the CM4AI consortium, it was really important to consider diversity associated with a cell line, the kind of cell line and its reliability. All of these factors needed to be considered. It also needs to be considered whether or not informed consent was involved. So below, we have two different squares here that point out all of these different considerations. And here you can see that one line on the right, the cancer cell line, consent was ambiguous, but it was actively chosen for diversity and inclusion reasons. Furthermore, you can see to the left, documented consent was available for the pluripotent cell line. Also important consideration. So on this slide, I just want to really emphasize that the idea of cell lines and informed consent is extremely well discussed in the LC and bioethics literature. And I'm not going to try and discuss it all within this slide, but just to make the point that within the CM4AI consortium, it was really important to us that the multitude of different considerations were given serious attention and balanced against one another. And again, really important to ensure that scientific validity is prioritized alongside other considerations such as diversity and inclusion, as well as consent. Next slide. And this slide is just to point out that good ethics really happens under a solid governance structure. The CM4AI data processing and access is governed by a data management committee designed by CM4AI leadership, and data ownership and licensing is fully described in the metadata. I won't go over all points in this slide, but just important to point out how much thought was put into this governance structure within the CM4AI consortium. Next slide. Just a few points to end on insofar as when we talk about ethics related to the CM4AI consortium, generalizability is an important point to consider. So was some kind of prioritization required in terms of what populations might benefit from downstream tools and the data generated? What specific populations may need this tool more than others? Or is the goal to make it as generalizable as possible? Should we ensure that data and tools are representative of a broader population or a specific disease? All of these are important considerations that were especially prioritized throughout the process. We also wondered who should have access to the data and who can utilize it. All non-commercial users can access through Dataverse, and commercial users have to separately negotiate, giving us more control over potential misuse, trying to control for that. Importantly, who benefits from this research? Beneficence. Scientists and scientific research broadly certainly does, and potentially patients through improved drug target selection. Another point to consider is what disease types were prioritized. Breast cancer, cardiovascular, neurological disease were prioritized, but the data sets and tools can be broadly applicable to multiple diseases. And lastly, trust and trustworthiness issues. We approach trustworthiness largely through our transparency and explainability goals, but to also emphasize that the literature is constantly evolving on the complicated nature between explainability and transparency and trustworthiness, but we nonetheless find it to be a worthwhile goal to prioritize trustworthiness, explainability, and also just ensuring that there is a lot of transparency in the data sets and within the metadata. All right, next slide.
Thank you, Danny. So for the last portion of our presentation, we'll go through some of the collaborations and partnerships that we've been developing through CM4AI, and also in collaboration with our other Bridge to AI partners and NIH collaborators as well. The goal of these collaborations and partnerships is really to increase the access to and use of the data and tools that we're producing as part of the Cell Maps for AI project. Some of the collaborations that we have developed and are actively engaged in include working with the NIH Common Fund Data Ecosystem for metadata integration and dissemination within CM4AI, as well as sharing some of the tools and frameworks, including Fairscape, with other grand challenges such as Chorus. We also have active collaborations with several Bridge to AI working groups, including Standards, ELSI, and the training work group, which is helping present this lecture today. In the broader community and outside of Bridge to AI, we're also participating in several standards-oriented communities, including the RO-Crate community and bioschemas.org. Next slide. Some of the work that we've done really within CM4AI is really focused on these three key points of improving, expanding, and empowering. One, we want to improve the number of things that we can do within the CM4AI program, including mentorship and career counseling, going beyond just training. And how do we really expand workforce and give opportunities to those who may participate in our programs? The next step is expanding and making sure that we have diversity in our early career technical pipelines and the individuals who participate in various CM4AI and Bridge to AI events. And our last step that we focus on is really empowering the grad students, postdocs, other trainees, and faculty to really leverage CM4AI and use the data and tools that we produce to their full potential. Some great work by Pam and Pam Foster and others in CM4AI has really expanded our collaborations and partnerships, including with several of the HBCUs, and work that we're trying to align with AIM Ahead to also host some of our events at specific sites to increase the access and accessibility to our training resources. Next slide. Really what our training focuses on is trying to bring people into understanding what cell maps are all the way through the other end of that AI pipeline. So as Trey and Tim both mentioned, we are really AI enabled from the beginning of our data generation through our data processing, building and assembling those cell maps, and then using the final tools to develop things like visible neural networks to predict things like drug response in cancer. Most of our academic focus or our training focus has been initially on really explaining what are these cell maps and what content and data are used to create them. Afterwards, we move into the tooling. Once we have those raw data, how can the cell maps actually be assembled using the tools made by CM4AI and other collaborators? And finally, how can we use those cell maps and actually apply that to human disease and precision medicine? The training opportunities that we've hosted so far, we've distributed virtually as well as in person using the Canvas platform, as well as a recent YouTube channel where we're able to distribute static content related to each of these steps of the cell map pipeline and final applications. Next slide. One of our first events was our 2024 Virtual Code Fest, and the emphasis of these first two events was really on leveraging the raw cell map data, so the mass spec, the immunofluorescence, and the CRISPR and Perturb-seq data, and really showing participants how to interact with those data sets directly, how you can visualize those data sets, assess for quality, and also start to look at protein-protein interactions or relationships. The next step of these training courses was then to take those raw data and actually run the cell map pipeline. So how do you go from the raw data sets and assemble a cell map that can be visualized and used in those other downstream applications later on? Our first event had a total of 38 registrants and 19 participants or trainees. 40% of those participants identified as female and 30% were from underrepresented communities. For our second event was hosted with Bridge to AI at our face-to-face open house on the NIH campus. This is an anticipated yearly event for people who may be attending in the future. We will be continuing to expand and progress the training that we offer during that event. And in 2024, we had another 29 attendees who attended the CM4AI specific track. One other exciting point from these two events is that we actually had several participants who actively contributed to some of our tools and code within CM4AI, and we're actually able to be on some of our poster presentations and likely on future manuscripts and research output as well. Next slide. And then our most recent event was our in-person internship. So this was a two-day event that we hosted in person at Yale University. We were able to cover travel awards to have 20 participants attend the event here at Yale, along with 6 investigators and instructors to help throughout the course. This event featured a combination of both synchronous and asynchronous didactic content with a key goal of trying to limit to less than 3 hours of didactic content over those 2 days. We really wanted to emphasize that hands-on training, make sure that people were able to use the code, tools, and data that we were providing, and also so that we could get feedback from them about how easy were our tools to work with and integrate and lead to actual output and science on the other side. For our participants, it represented 6 academic, 1 healthcare delivery, and 3 industry organizations with a broad range of experience and backgrounds, ranging from master's students to early and later career faculty. Next slide. The output of this event, this was hosted in person. We provided virtual environments and compute for everyone to work with the data. Day 1 of the event was focused on building out a visible neural network. So how can you take the cell map, train that visible neural network on actual drug response data in cancer and other disease, and actually find which cellular pathways are responsible for those cells responding to the drugs that were given. On the second, we leveraged the output of those VNNs once we actually had the cell map and different gene sets to use large language models to annotate and say which cellular pathways and complexes might be involved or were contained within a specific community within the cell map. Over those 2 days, the 20 participants ended up running over 1,400 AI and machine learning experiments, so it was a very productive event. And prompting challenge for all participants and integrating that into an MLOps pipeline. So rather than just running the code locally and looking at results in a text file, actually integrating this to a web interface where you can see those results live, track your experiments, and tie in that next layer of explainability. So as Tim mentioned, a lot of our work initially focused on that pre-model explainability. Where did the data come from and what was the metadata for the data assets? In this step, we're actually moving into that in-model explainability, which hyperparameters are used, which data sets were used, and what was the training performance for all of the outputs. Next slide. As we move into year 3 of our Bridged AI and CM4AI award, we've got several different things that we're looking to continue to expand the training opportunities and access to those opportunities for future participants. One of the events that we will be doing is hosting an in-person training event at an external site so that we can engage as broad of an audience as possible and also decrease the barrier to attending where participants don't need to travel to another site. We are also increasing the amount of asynchronous and hands-on learning opportunities that we can provide. The in-person event that we just hosted, we are actually converting both of those day events into virtual events that can be done as a learn on your own. And we will be planning to host the actual challenge components in either Synapse or Kaggle. So as people fine-tune their models, engineer better prompts, they can actually compete and see those on a leaderboard for looking at what the results are and how changes to the models can impact the downstream performance. One other thing that we're excited to engage with the community on is also quantifying what is the impact of these events. So getting feedback from all of you of how valuable was the presentation today, if you have attended or attend our events in the future, really looking to get that feedback so that we can make sure to tailor content to what people need and want to be able to most effectively use the data and tools produced by CM4AI. And we also look forward to having further integration and expansion of these training events with the Bridge Center and our other grand challenges as part of the Bridged AI program. Next slide. And just to really summarize where we're trying to go with this, we've talked about the visible neural networks, the large language models, the cell maps, but this end goal is really being able to take that cell map architecture that Trey had walked through and be able to really apply that to precision medicine and biomedical research. And one example of this was the drug cell paper published by the Ideker lab, where that cell map architecture is actually used to then generate this neural network architecture. So instead of having a fully connected neural network, it's partially connected based off of the actual structure of the cell. And when genomic data, so in things like genomic mutations or copy number variations are input into that model, it can be trained along with drug structure embeddings and then actually predict what is the expected drug response for a specific sequence. The benefits of taking this visible and explainable approach is that down on the bottom, we can actually then track back through those cell map architectures to see which pathways are involved and actually use that to identify new targets, explain which things are, lending the most weight to the predictions, as well as guiding future biomedical research studies of are these predictions correct, as well as are the cellular networks that we've designed correct as well. With that, we'd be happy to take any questions, and otherwise, Trey, I can hand it back over to you for any final summary or comments.
No, that's a wrap. Do we take Q&A or Jack and Pepe, maybe you can give us some guidance.
Yeah, definitely. Thank you, Dr. Ideker, Dr. Clark, and Ms. Pacia and Dr. Schulz for your insightful, engaging presentation. Your highlights are offering everyone in depth and a sense of the depth of the collaboration within the Bridge to AI initiative. And we deeply appreciate the time and expertise you've shared with us today. We do encourage people to ask questions. I think I did see a question a moment ago. Let me read it out loud. What level of programming experience is required to participate in the Code Fests? Will there be another one in the future? And will there be any guidance or resources provided for beginners during the event?
Yeah, it's a great question. And so we've had a really wide range of experience for people who have come into our events. I would say at least a basic understanding of Python does really help for being able to quickly get through and start to work with the code. With that said, the tools, especially as they are wrapped and relatively easily installable, if the goal is to just run the cell map pipeline, see the outputs, and work with those outputs to answer other scientific questions, that requires very minimal prior coding experience to work on. And so we really welcome people from any background, but do encourage at least some basic understanding of Python to get the most value. But happy to discuss with any individual participants of which events may be best, or if there are other online trainings offered by other Bridge to AI data generation projects or other locations and training resources that could make you more prepared for the various events that we do offer.
And just to add to that, Jack, I don't know if there's a way to link some of the external training resources Wade mentioned to where this video is posted. Certainly, you can access them through the cm4ai.org URL shown there at the lower left or lower right. One interesting aspect about this project that I think we're seeing more and more in genomics and biomedical research as a whole is this so-called multimodal aspect. So you have multiple modalities of data, which 10 years ago would have been treated entirely separately with separate learning pipelines. So protein imaging was and is an entire subfield with educational resources and training and ML approaches that are applied to that data stream alone. Similarly, protein mass spectrometry is really a large subfield of biomedical research, which again has separate training resources that one would have studied previously. What is now happening with these integrative AI-enabled approaches is as you begin to put all these different types of data together, you need integrated training programs that cover all of them in moderate depth. And that's really the opportunity that Wade has really taken on here.
Excellent. Thank you. We do have another question. This is from Shurjo Sen. Can you place CM4AI in context of the larger unsolved puzzle of decoding the human genome? Will it be one step in a 100-mile journey or more like half a mile?
Yeah, certainly not more than half a mile towards the 100-mile journey. But I would argue it's closer to half a mile than a step. I think the key word in understanding where we fit in to genomics as a whole is interpretability. And so you can think about there's lots of approaches that are connecting changes to so-called genotype, to your DNA, directly to changes to so-called phenotype, how you present as a person, whether you have heart disease or cancer or you do not. If you think about direct efforts, which have 90% of the journey thus far, if you want to look at how many miles we have come so far, Shurjo, looking backwards, 90% of the path has been direct efforts to connect changes in nucleotides to changes in disease. And if you think about it, that's bridging a very, very large gap, a change in a nucleotide. We might consider this at the nanometer scale, at the size of an ACG or T, as it were. Whereas a change in a person, of course, is happening at the meter scale. And so we're really asking these approaches previously to bridge those 9-10 orders of magnitude. What CM4AI is trying to do is insert what happens in between. What are those molecular and cellular pathways by which variants and mutations affect molecular pathways and complexes and cell types and tissues and so on their way to influencing phenotype. We've, of course, seen some of that work done already through efforts like the NHGRI and their extramural research community. Here, we're really targeting cell biology as the missing gap for understanding how to convert the genotype and the genome into phenotype.
Next up, thank you, Dr. Ideker.
So I think even though Tim has already typed in the answer, Tim, could I trouble you to share that answer with our entire audience? We have a question from Dr. Haluk Reset.
Right.
Jack,
Read the
want to
question.
read the
I'll
question?
respond. Okay.
The question is as follows. It is great that you are using Dataverse Gray, GREI. Can you briefly discuss how your experience has been so far with data and metadata deposit, etc.?
Yeah, so I actually have a long collaborative background with the Dataverse people from when I was at Harvard, and they're great. We built a push button interface to their API so that you can and you can go to basically any Dataverse instance in the world now if you wish and deposit that our data but directly from Fairscape. Now, an issue that comes up though is that Dataverse as a generalist repository is kind of restricted, or searchability, let's say, is restricted to the DataSite metadata schema, which is sort of like bibliographic metadata. And we provide a lot richer, far richer metadata, and we'd like to make that searchable. So I've been discussing with Stefano Iacus, who's the Dataverse program manager, about how to do that. And we have some ideas, but we have to track down some funding and so forth to be able to expand that searchability.
Thank you, Tim. Haluk, does it answer your question? Is there a follow-up question?
Yeah, and let me add one thing, Haluk. These guys in Dataverse are very interested in doing this, and they're very amenable to working, very cooperative, I'll say. Right.
But clearly, Tim, this process and tool you have created that's automating the metadata annotation and creation, this is really important. Is that an approach you are sharing with the other GCs at Bridge to AI?
Yeah, I have had a number of conversations with, and we've done some experiments with the Chorus Grand Challenge. I think again, this is a little bit funding limited right now, but we are definitely interested in sharing it with the other GCs, and I think it can be. There's some technical things that we have to solve, but we know how to solve them. And it's just a matter of putting some engineering resources on it.
Fantastic. So next question, Jack.
I think Jack dropped off.
Let me read it. It's we have next question from Ian Stevens, what is the present and future role of an ethic person in the setting of basic science research? Are they more of an embedded scholar to consult or an extension of the IRB? Interesting. Trey and Danielle.
Well, I'll give Danny the first dibs at that one, if she wants to.
Yeah, I'll start answering and then anyone else can jump in. What an interesting question, because I think it speaks to a lot of structural barriers to that happening. Typically, that isn't how funding is structured. In other words, it's so rare that you come across an opportunity like Bridge to AI where this level of collaboration between ethicists and basic scientists can happen. But I will say increasingly in the literature, at least from my perspective, I see it as an increasing interest in the field of LC and ethics. So for so long, the focus has been primarily on research that's more directly related to patient populations. But I think people are becoming increasingly aware that basic science actually does have moral implications associated with it, with value-laden decisions that people may see as neutral, but nonetheless actually have a lot of downstream effects, such as disease prioritization or some of the really nuanced balancing and informed consent conversations that we were having in the presentation. But I'll end by saying that one example that I see that I've always found really interesting is George Church's lab at Harvard and MIT has an embedded ethicist that provides advice, consultation at a lot of the stages of the basic science research. So I'll just point to that example and just end it by saying, I hope in the future there can be more collaboration, but right now I think the conversation about the future of such collaboration needs to be focused on the structures that will allow those kinds of collaborations to flourish. I'll turn it over to my other panelists in case they have opinions as well.
I mean,
Trey
I thought that was a great answer, Danny. I mean, if I was to add anything, it would be a complementary point that well, first of all, I think that part of what happened in the Church lab is I'm imagining the goals from NIH staff as they developed the entire Bridge to AI program is to embed the ethics teams. One way I think in the CM4AI project that is we're basically trying to embed the ethics in the AI through interpretable or explainable machine learning. Now, that opens up a large debate. To what extent does being able to explain the inner workings or the decisions that an AI has made provide ethical support? That's a very interesting debate, but that's the direction we've taken in our DGP project.
Amazing, Trey. I think the innovation part that your project has contributed, really a pioneer work, is to automate the process of how to involve ethical considerations in the data design.
That's right, and certainly to increase trust, to increase understanding, and through understanding to increase trust. When you understand what are the molecular rationale by which a model predicts a successful drug response or a drug resistance outcome in the case of the example Wade gave at the end.
Yes, thank you. I have a follow-up comment from Haluk, Dr. Reset. So let me read this. Searchability limitations is a common issue that we keep hearing about over and over. Hopefully, it will be expanded. ODSS is working on it too. So I guess I share that with Tim. Any thoughts, Tim?
Yeah, I think what we are thinking about is actually making is actually schematizing the RO crate packages that we send so that they can be typed per the kind of project, and then essentially glued into whatever generalist repository you wish and add it as a search index. That's kind of our model for this. And we're going to work up a proposal on that.
Thank you, Tim. I want to thank our audience for participating in this very important lecture introducing AI-ready datasets, its structure, its protocol, its guideline of how to accomplish AI-ready and trustworthiness. Thank you again for joining all of us, and thank you all for our esteemed speakers. Have a great day. Bye-bye. Thanks so much, everyone.